Hi everyone, welcome to Unit 7, Chapter 4, the last lecture before the end of the semester. And while I'm sad to see everybody go, I am really excited to teach about the nervous system, which is my favorite system to talk about. And we'll also be learning a little bit about the special senses. So, what makes up a nervous system? Well, a nervous system is going to be a network of specialized cells which can send and receive signals. So, when we're talking about very rudimentary organisms such as the hydra or the jellyfish in the family Cnidarian, we're really just talking about a nerve a net of nerves. It's very net-like organization with crisscrossing pattern of nerves, and they're not really there to sense the environment as much as they are there to coordinate movement from contractile filaments inside of the organism. So we'll call these epitheliomuscular cells. They have a little contraction in them, and this nerve net allows them to coordinate those contractions. Once we get to the planaria, we have some advancement of the nervous system. It's now ladder-like, showing a more consistency throughout the body of the organism, but also cephalization. You see that we start to develop a specialized head with sense organs such as the eye spot and a brain, or at least a integration center called a ganglia. So that's what we start to see with the planaria. Once we get to higher order animals like arthropods, mollusks, and vertebrates, we continue to see that cephalization where we have all of our sensory organs kind of focused in one spot, with our integration area, the brain, also located in that one spot. Now, with these complex animals, the nerves get more and more specialized and are able to do more and more things, such as sense things. So we have specialized sensory organs like the antenna or the eye, which can help us react to the environment around us, which is the whole point of the nervous system, is maintaining homeostasis. Now, how do we do that? Sending and receiving of signals with the neuron. The neuron is going to be a specialized cell that is only found in the nervous system, and it is completely developed just to send and receive signals. Now, neurons still have to contain the normal organelles that all cells need, such as a nucleus or even the endoplasmic reticulum and the little ribosomes. Those are all still there. But in addition to that, we have some specialized organs in order to send a signal. The main specialized organelle is going to be the axon, which is this really long branch off of the main body of the cell. And you can see here in this GIF, that is where the signal is being sent. So we have two cells here, or two neurons here. One is sending the signal this direction, and one is receiving the signal. We call this a presynaptic cell and a postsynaptic cell. The postsynaptic cell is after the synapse. The synapse is going to be this very, very small location where the signal is actually being transferred from one cell to the next. So that's where we get the presynaptic cell and the postsynaptic cell. Now, at the end here at the synapse is the axon terminal, which is the end of the axon. The beginning of the axon starts at the axon hillock. You can see it here, where all of the signals from the other neuron are kind of collected or funneled to this one area called the axon hillock. And if the signal is strong enough, it will fire another action potential. If it's not strong enough, then no signal will be fired. So that is the purpose of the axon hillock. Next to the axon hillock is the body of the cell, where you will find all of the normal parts of the cell, such as the nucleus and those ribosomes. This is called the soma. The body of the cell is also called the soma. So it's called body, but it's also called soma. We'll see that in the next slide as well. Attached to the soma will also be something called dendrites. These dendrites are there to receive a signal. Here you can see another diagram of the same thing. Here you see the soma attached to the axon hillock, which is attached to the axon, which ends at the synapse at the axon terminal. Here we have the presynaptic cell, and here you can barely see the postsynaptic cell. And then attached to the soma is going to be the dendrites, which are there to receive the signal. Additionally, this diagram has something called myelin, which is an insulating, like fatty deposit around all of these axons. So we'll talk about the myelin in a little bit. Now, neurons aren't just shaped like this. This is a very standard shape of the neuron, but neurons come in many different shapes and sizes. All of them will have a soma or a cell body, and then they're going to have branches coming off of that. And the branches could either be dendrites or axons or a mixture of both. And you can see that these cells are specialized based on their function. So these olfactory neurons, for instance, they are specialized to receive smell sensation, and then they send the signal to these integrating neurons. These are the basic shapes of neurons. We recognize this one right away because this is what most diagrams look like, but this is actually only a specific type of neuron called a multipolar neuron, a pole meaning an end. And this neuron indeed has many ends, hence the word multi. So we measure the poles of a neuron based on how many branches is coming off of the soma or the body. Here we have 1, 2, 3, 4, 5, 6, 7 branches coming off of the main body, which qualifies it as multipolar. Here we have unipolar because if we look at the soma or the cell body, only one branch is coming off of this unipolar neuron. We also have a bipolar neuron, which of course has 2 branches coming off of it. And then interestingly enough, one of the more common types of neuron is actually a pseudo-unipolar. Unipolar because it only has 1 branch coming off of the main body of the cell, but that branch then branches off into multiple directions. So pseudo-unipolar is also a classification of a neuron. So in the nervous system, we have 2 main divisions. The central nervous system is, of course, central. It goes down the middle of your body and includes the brain and the spinal cord. Nothing coming off of that, though, just the brain and the spinal cord. You'll find that there are many differences in the central nervous system as opposed to the peripheral nervous system. The peripheral nervous system is anything not included in the brain and spinal cord. And so this is where a lot of your sensation is going to come from. In fact, the nervous system has 3 main jobs. Its job is to receive sensory input or reception. And that's going to happen in the peripheral nervous system. Imagine those touch nerve endings for touch. You're receiving the signal from your peripheral nervous system. And then your job is to integrate that information or to perceive the information. You don't always perceive every message that you receive. So this is going to happen in the central nervous system where you're going to be thinking about the information that you received. So perception. Are you able to perceive it? If it's not a very strong signal, you may not perceive it, but this is going to happen in the central nervous system. Imagine even pricking your finger on a needle or touching something hot. You don't necessarily think about it in your brain, but you do have integration areas where that signal is sent to your spinal cord, and then you're sent a secondary signal so you can think about it, but before that even happens, you send another motor signal to move your hand out of the way of danger. And that's happening here in the spinal cord. Still integration, still happening in the central nervous system. The last step is what we just spoke about, some sort of response, an output, and usually this is a motor output. the response. Either we need to say something by moving the muscles in our mouth, or we need to move our hand out of the way or move in some other way. We are going to respond to the stimulus, usually in the peripheral nervous system. So, what are some other differences between the central nervous system and the peripheral nervous system? Well, one of them is glial cells. Glial cells, glial is just the support cells for any neuron. And this is different in the central nervous system and the peripheral nervous system. In fact, these glial cells are so common. In fact, they outnumber neurons 10 to 1. There's way more support cells than neuron cells. That means that there's more opportunity for them to mutate. So most tumors are caused by mutations in glial cells just because there's so many more of them. And also glial cells are going to reproduce faster and divide faster than true neurons, which are notoriously slow to repair themselves. So in the peripheral nervous system, we have Schwann cells. Now, Schwann cells are specialized cells that produce myelin. And Schwann cells in the peripheral nervous system wrap themselves around the neuron, kind of like a burrito. And so that means that the nucleus and all the parts of the Schwann cell are kind of laminated down underneath all of this myelin. So they're here insulating the neuron. We also have something called satellite cells. These satellite cells do everything for the peripheral nervous system. They're going to provide nutrients and structural support to make sure that the neurons are located where they're supposed to be located, that they're insulated, that they have enough nutrients. Now, the central nervous system is a lot more crowded, a lot more neurons and more connections and more integration. So they need a little bit more support, starting with oligodendrocytes. Oligodendrocytes also make myelin, so don't get them confused with Schwann cells, because Schwann cells can only myelate one axon at a time. Whereas oligodendrocytes, everything is so crowded that they can touch multiple neurons at a time, and they can myelinate multiple neurons at a time. So the oligodendrocytes are responsible for the myelin only in the central nervous system. They also have astrocytes. Astrocytes behave a lot the same way that satellite cells do. They're going to provide nutrients and structural support. And they're called astrocytes because they kind of look like stars. A little star-shaped astrocyte there. Don't forget, we also have ependymal cells. Ependymal cells are very, very important because they produce cerebral spinal fluid, which helps to cushion and protect and clean the neurons. We also have microglia, which acts as kind of like the cleanup crew, and they will take away any dead or dying cells that might be cluttering up your central nervous system. So it's great that we have all these different support cells for the different parts of our nervous system. Now, why do we need myelin in the first place? Well, much like the mouse cord to your computer mouse has a rubber coating around it, it's important for neurons to have this myelin sheath. It helps to protect the neuron, but also to make the signal stronger and faster. The signal that's being sent down an axon, very similar to the mouse sending an electrical signal, is a chemical electrical signal. So we have ions lined up on the inside of the axon and ions lined up on the outside of the axon. These ions have to very quickly switch places when an action potential is taking place. By insulating the axon, it prevents these ions from switching place just anywhere, and now they can only switch place at the node of Ranvier, or this little gap in the myelin. This allows the signal to jump from section to section, moving much faster and also not losing as much energy in the process. This is what it looks like in a GIF, where you can see that this top axon is myelinated and the bottom axon is not myelinated. You can very easily see that the myelination causes the signal to move much faster, which is very important for your cognitive functions, because if you have a disease that attacks the myelin, it can cause cognitive decline. Common demyelinating disorders are like multiple sclerosis or MS. This affects the myelin in your oligodendrocytes, so your central nervous system. It can cause weakness and vision problems and difficulty with balance and coordination. Guillain-Barré syndrome actually attacks your Schwann cells, so it is going to attack the myelin in your peripheral nervous system, which can lead to muscle weakness, tingling sensations, and in some severe cases, even loss of mobility. Now, I said that the inside of the cell and the outside of the cell are lined with ions. You would think that some of the ions would be negative and some would be positive, but that's not the case. We're mostly going to be talking about sodium, which is a positive ion, and potassium, which is also a positive ion. What we have in our cells is a ionic gradient, meaning that we have a much stronger positive charge outside than inside. We are constantly pumping positive sodium outside of the body or outside of the cell. Okay, and we have a little bit of positive ions inside with the potassium, but mostly we're talking about sodium being outside and being highly positive. We do have potassium inside as well, but there's just not as much of it. So we end up with most of the positive charge being on the outside. Then during an action potential, remember that we mentioned that if the signal gets strong enough, if this signal at the axon hillock is strong enough, it will fire an action potential. Well, for an action potential to take place, the signal at the axon hillock has to reach a certain voltage. And once that difference, that membrane potential voltage meets, then these gated ions will open up, allowing the ions to rush back into the cell. So let's look at what that looks like laid out. So we are at our normal resting potential. That's where we're going to start out with. Number one here, we're at our normal resting potential. We might get some excitatory signals, we might get some lower signals, but once we reach this point, negative 55 millivolts, we've reached the threshold of the action potential, which means it's an all or nothing after this. We are going to go through a process of rapid depolarization. The reason we go through a process of rapid depolarization is because that we've met the threshold to open the sodium ion channels. Now that these channels are open, sodium can rush into the cell. It's rushing into the cell, rapidly raising the charge of the membrane potential until we reach about positive 40. At about positive 40, the ion channels close for sodium, but they open for potassium. So now potassium is going to start flowing out of the cell rather quickly. This works to repolarize the cell, and it works so well that we end up in an area called hyperpolarization. Once we end up in this area called hyperpolarization, we're really too polar to initiate another action potential right away. I want you to think about the toilet, and after you flush the toilet the first time, you kind of have to wait for the toilet to reset itself, to refill, before you can flush it again. That's what this area is representing. The hyperpolarization state is overly negative, so that a new excitatory signal that normally would reach the threshold at the resting potential is not going to be strong enough to reach the threshold now. So we're not going to get another action potential right away until we reach the resting state. And the way that we get back to the resting state is with a special protein called the sodium potassium pump. The sodium potassium pump works constantly to push sodium out of the cell and to pull potassium into the cell. And we're only pulling in 2 potassium for every 3 sodium. So there's going to be a lot more positive charge outside the cell than inside the cell. And also, potassium can leak through the membrane and sodium can't. So we're going to lose some potassium naturally, which further separates the pressure gradient more positive outside than inside. So all together, we have a threshold. Once we've reached the threshold, we will go into rapid depolarization because sodium ions are flooding in to the cell. Then once we reach positive 40, we close those channels and open up the potassium channels, and now potassium is going to flow out of the cell until we get to hyperpolarization, and then everything is up to the sodium-potassium pump, which does take ATP. So we're going to use energy to go back to the resting state. If you'd like to know more about how these action potentials take place, you are welcome to watch this video, which is a great introduction to an action potential and how the action potentials work. Now, what happens once that action potential takes place? Well, that action potential will send a chemical electrical signal down the axon until we reach the axon terminal. The axon terminal ends at a place called the synapse or the synaptic cleft. See, the neurons don't actually ever touch each other. Much like a child in the back seat of a minivan saying, "I'm not touching you," they're just barely not touching. So synaptic cleft does not touch. What we have is we have the presynaptic neuron sending a signal to the postsynaptic neuron. So the postsynaptic neuron has to have a few things. One of those things that it has to have is it has to have a receptor, something to receive the signal. And that receptor has to match the signal being released, called the neurotransmitter. Now, as we learned previously when talking about hormones, what qualifies a material as a hormone is not what it's made of, but how it's being used. So the same thing applies to the neurotransmitter. What the neurotransmitter is not as important as how it's being used. The same molecule in your brain acting as a neurotransmitter might have a different effect in the rest of your body. So a common neurotransmitter might be dopamine or norepinephrine even though those have different purposes in the rest of your body. So what happens when that signal reaches the synaptic cleft? Well, it actually triggers a cascade of events. The first of which is releasing extra calcium. So that chemical electrical signal triggers the release of lots of calcium, and calcium causes a shift in the polarization of the cell, which causes these vesicles to just be rejected. Now, these vesicles are kind of accumulated by the cell. It's constantly making and packaging these vesicles, and they're just kind of waiting around for a signal. That signal is the calcium being released. It tells this cell to reject all of these vesicles. That means that our synaptic cleft is going to be flooded with neurotransmitter. And again, it doesn't matter what kind of neurotransmitter we're talking about. We could be talking about a dopamine neurotransmitter. So now we have tons and tons of neurotransmitter able to activate these receptors. So now the receptors are activated, sending a signal to the postsynaptic cell. Once the signal is sent, several things could happen. If we are no longer receiving the signal down the axon terminal and we're no longer releasing calcium, what the cell will actually do is revert back to its normal activity of packaging neurotransmitters inside of vesicles. So we can do that by making new neurotransmitters, or we can just recycle the old ones and take up any old neurotransmitter and repackage it into a new vesicle. That way we clear out the synapse and we save energy by repackaging a neurotransmitter that's already made. Another process is probably happening at the same time called enzymatic degradation, where an enzyme is kind of coming in and breaking apart these neurotransmitters so that they can no longer activate the receptors. Now, neurotransmitters are very, very important for your body and having the appropriate amount and balance of neurotransmitters. For instance, the neurotransmitter dopamine. Not enough dopamine can cause bradykinesia, or very slow movement. This is one of the hallmark symptoms of having Parkinson's disease. This is a degenerative disease that attacks dopamine-producing neurons, so you don't have enough dopamine in your brain, causing that slow, jerky movement. Now, schizophrenia, on the other hand, is an overproduction of dopamine, and this contributes to things like hallucinations and disorganized thinking. I find it very interesting that the treatment for schizophrenia, which is oftentimes fake dopamine or L-DOPA, can cause Parkinson's type symptoms after long-term use of L-DOPA as an intervention to schizophrenia. Drug abuse also can target different types of neurotransmitters by either increasing or deregulating the release of that neurotransmitter. If you'd like to know more about these illicit drugs and their effects on your brain, you can click on this interactive of these mice. Now, this is not testable information, but it is a fun little game where you can choose the different mouse that is, using a different illicit substance, and then you can analyze them and learn a little bit about the neurotransmitter being affected by that drug. So that's just a little interesting side quest. That brings us to the cerebrum. So we learned about the neuron. Let's learn about the cerebrum as a whole. This is the large portion of what you think of as your brain. It's made up of two hemispheres, the left and the right hemisphere, with a long division down the middle called the longitudinal fissure. But your left and right sides need to talk to each other, so we have the corpus callosum. It's almost like a little strap of axons. So these are just for sending signals back and forth between the left and right brain. The left brain tends to control more analytical verbal functions, but also controls your right half motor functions. The right side of your brain controls more creativity type functions, but also controls your left motor functions. So any damage to the right side of your brain will have left-sided mobility issues, and any damage to the left side of your brain will have right-sided mobility issues. This can further be seen by experimentation of patients who have had damage to their corpus callosum. Remember, the corpus callosum is the communication between your left and right sides. So if I show a patient with damage to their corpus callosum the word face, they may not be able to verbally tell you anything about that word, but they might be able to draw a picture of a face. If you show them an image of a face, if you show them an image of the face, they may be able to say the word face, but they wouldn't be able to draw it because of that separation between the left and right hemispheres. Now, the cerebral cortex is going to be made up of gray and white matter. As we mentioned, the corpus callosum is a bunch of axons. That indicates that it is going to be white matter because that white coloration is due to the myelin surrounding the axons. The cell bodies, however, do not have myelin and appear more gray. So we have these integration areas where the cell bodies are located along the outside of your cerebral cortex, whereas transmitting of signals is deeper and is more of a whitish coloration. So the human brain has several major parts, one being the diencephalon. The diencephalon is this part of the brain, and this is going to be your basic functions. This is sometimes called your lizard brain because it's your most basic part of your brain. If you had damage to your cerebrum, you would have significant loss of functions, but you would still be breathing and you would still experience thirst and you would still have a sleep-wake cycle because all of those things are controlled by your diencephalon or your brain stem. In fact, your cerebellum or your little brain right here is mainly for coordination and balance, whereas your pons in the middle helps with coordinating breathing. This young man had damage to his pons in his brainstem, and he actually can forget to breathe. Your breathing is controlled by a semi-voluntary thought happening in your brainstem, and it makes sure that you breathe while you're sleeping or distracted. You can't force yourself to not breathe. So unfortunately, this man had damage to this area, and now during wakeful hours, he is able to remember to breathe, but during sleep hours, he has to wear a sleeping mask to force air into his lungs because he would forget to breathe due to the damage to his brain stem area. So the brain stem is made up of these different parts in the diencephalon, the pons being one of them. Your thalamus and hypothalamus located up here are more for your thirst, wake cycles, those involuntary urges, whereas other parts of your brainstem have to do with vomiting, coughing, sneezing, hiccuping, like those semi-voluntary things. So your cerebrum has several different functional areas. They're usually divided into lobes, and within those lobes are further functional areas. So first is the parietal lobe. The parietal lobe is on the top of your head. You have a left parietal and a right parietal, and this is more of like your VR center. This helps you to interpret the world around you and create like a virtual reality for yourself. This is also where a lot of logic and arithmetic kind of comes in, making sense of the world. And then we have the occipital lobe. The occipital lobe is located in the back of your head, and this is also where your visual area is. It's kind of right here, your visual integration area. Underneath that is the cerebellum, which we already mentioned is a point of balance and coordination. Underneath that is the brain stem. Remember, breathing, sleep-wake cycles, thirst, hiccuping. Then we also have the temporal lobe. The temporal lobe is kind of folded over the top of the frontal lobe, which is the largest lobe. This is an area that's associated with learning and memory, but also language. All right, the frontal lobe. The frontal lobe does double duty. It's going to take care of all of our decision making. So thinking about moving your muscles is a huge part of that. Moving the muscles of your body is a huge area that's located within the frontal lobe called the motor cortex. It's important for movement. And it is right next to the sensory cortex, which is important for sensing things. That's part of the parietal lobe. But these two work together to create the somatosensory cortex that helps to coordinate sensation and movement. The frontal lobe also does things like emotions, behavioral control, personality, executive function. Let's see. So we have all of these different functional areas, and it would be good to learn what those functional areas are. And remember, there are functional areas on top of functional areas. So for instance, the occipital lobe is this whole area, and it is mostly associated with vision, but the primary visual area is this smaller area on top of it. So how do we know that these different areas work for these different things? Well, we know that based on damage. One such study is the study of Phineas Gage. Phineas Gage was a construction or a railroad foreman, which means he's in charge of a bunch of people building a railroad. And one of the things that they did was they used TNT, a highly volatile explosive. And they would dig a little hole and push the TNT into this hole. And if they weren't satisfied with how deep the TNT went, they would take a large rod called a tamping rod, and they would smash the TNT into that hole or tamp it down. Obviously, this was extremely dangerous because one day when Phineas Gage was tamping his explosives, a spark ignited the explosive and threw the rod up and through his face. Damaging his eye and the top of his skull, taking out this part of his frontal lobe. So this whole part was gone. Now, he did survive this injury. Miraculously, he survived, and he actually lived a long, relatively normal life afterwards, except that now he had problems with impulse control, alcoholism, anger, all of those charming personality qualities that gave him his title of foreman, all of that was gone now. He's no longer able to, command a team. So we assume, based on Phineas Gage, that this area of the brain must have been really important in impulse control and decision making, because he was no longer able to do those things. Also, there are these interesting little areas. If a person has a stroke in this area, this is part of the frontal lobe, not part of the temporal lobe, but it's very close to the somatosensory cortex. In fact, it's close to the area that moves your mouth. If you have a stroke or damage in this area, you will no longer be able to speak. This is called expressive aphasia, because you will not be able to coordinate the movements of your mouth properly if you have damage in this area. So you may be able to understand someone speaking to you, but you would not be able to speak to them because you wouldn't be able to move your mouth properly. Now, if you have a stroke in this area, it's kind of partially part of the parietal lobe, but mostly in the temporal lobe. This has a lot to do with interpreting and understanding speech. So if you have a deficit in this area, this is called receptive aphasia, where you might be able to formulate words in your mouth and they might make a degree of sense, but somebody speaking to you would sound completely foreign, even if they're speaking to you in a language that you've spoken your entire life. You will now have Wernicke's aphasia. So this is another way that we can map different functional areas of the brain, is by studying case studies where there have been damage to those areas. Still talking about the central nervous system is the spinal cord. Now the spinal cord is kind of opposite of the cerebrum, because the spinal cord has white matter or myelinated axons on the outside and has gray matter or integration areas on the inside. And the spinal cord is actually where reflexes take place. So there's something called a reflex arc. A reflex arc is, let's say, is a pair of nerves. One is afferent and one is efferent. Afferent means the signal is coming in. So let's say that you touch something really hot. That signal is going to come in and be integrated in the gray matter of your spine. And we're going to immediately say, wow, that's hot. Let's move our hand out of the way with this efferent motor neuron. So this is going to be a motor neuron that tells your brain, or tells your arm, to move out of the way because it's hot. Now we're going to send a signal up to the brain so that your brain can also be keyed in and say, I just moved my hand because that was hot. But you're not really going to get that signal ahead of time. You're going to almost think about it after the fact, after you've already moved your hand out of the way. And we're able to do this because your spinal cord does have gray matter, does have those interneurons. So these reflexes are rapid, very predictable, involuntary responses, but they're still considered somatic because they have to deal with skeletal muscles. And they're only going to occur in one direction. Like our reflexes are pretty predictable. If I touch something hot, I'm going to pull my hand backwards. I'm not going to start doing a dance. It's a very predictable pattern. And autonomic reflexes are going to deal with things inside of your body, whereas somatic reflexes deal with skeletal muscle. So here's a better picture of that pinprick. Let's say that our skin receptors feel that painful stimulus. They're going to send a very quick signal to this interneuron, which is going to do two things. It's going to tell this motor neuron, move your hand out of the way, and it's going to send a secondary signal to your brain to say, hey, we moved our hand out of the way. We have two different systems. We already mentioned it when we talked about reflexes, autonomic reflexes versus somatic reflexes. That's because somatic has to deal with your skeletal muscles. Anything to do with your skeletal muscles is going to be considered somatic. Now it says conscious activity, kind of. Okay, I'm not necessarily conscious that I'm moving my hand away, but it still is dealing with skeletal muscles. Autonomic, on the other hand, is going to deal with anything inside of your body, your smooth muscles and your glands. So autonomic is your automatic system, the one that you don't think about, whereas the somatic system, you think about it a little bit more. Now, the autonomic system has two divisions, the sympathetic division and the parasympathetic division. The parasympathetic division is going to be your rest and digest, whereas your sympathetic is going to be your fight or flight. That means they usually work opposite of each other. They're going to affect your internal organs, but the sympathetic system is going to get you excited. It's going to increase your heart rate and decrease unnecessary things like digestion. Whereas your parasympathetic is going to increase things like digestion and decrease things like heart rate. So these things are working in opposition to each other. But they also use different nerves. For instance, the parasympathetic, or your rest and digest, is mostly going to be using cranial nerves and sacral nerves, whereas your sympathetic, or your fight or flight, is using mostly thoracic nerves. And so again, these are going to be excitatory, whereas these are probably going to be more calming, except for the digestion. We're going to activate digestion with the parasympathetic. Here's a better image. Again, you can see where most of the nerves in the parasympathetic come from cranial nerves, or nerves coming directly off of the brainstem, and sympathetic is happening mostly in the thoracic area. So when we are sensing the world around us, we mentioned that we have to have reception, perception, and then response. So there has to be some sort of stimulus. We receive the signal, we perceive the signal in our central nervous system, and then we respond to it. And this all has to do with ion channels being affected. So we have different receptors in our body that are going to cause these ion channels to be activated or deactivated. For instance, the receptors in your eyes respond to light. So they are photoreceptors, and they are going to start triggering an ion channel effect with the presence of photons or light. So this process can be adapted, or if there's too strong of a signal, your body can protect itself. So there's two things that happen: sensory adaptation. Sensory adaptation is kind of like when I first put on my socks in the morning, they feel really tight, but then after being in them for a couple hours, I stop noticing. That is sensory adaptation. You're going to adapt to a stimulus that is happening constantly, and your body is overwhelmed by it, so it just kind of starts to ignore it. Remember, your body is constantly being bombarded with this sensory information, but you tend to ignore most of it unless it is attention grabbing. So for instance, if I stick my hand in a cold glass of water for a couple minutes until I adapt to it, and then I move my hand to a room temperature water, that room temperature water is going to feel really warm even though it's not, because I've adapted to the cold water. Whereas habituation is more adjusting to a habit or something that you are exposed to frequently. So whereas sensory adaptation is constant stimuli, habituation is frequent stimuli. For instance, I frequently go eat ice cream after class, and eventually after eating ice cream after class every single day, the ice cream just doesn't taste as good to me anymore because I'm too used to it. It's no longer a treat. So we talk about habituation a lot in drug addiction. So what kinds of receptors do we have? We mentioned photoreceptors for vision, but we have many different kinds of receptors. We have special cells that act as chemoreceptors. They receive smell and taste and chemical information. We have specialized mechanoreceptors that are mechanical. They actually perceive mechanical changes, such as taste, touch, and hearing. And we'll talk later about how is hearing a mechanical sensation, but it is a mechanical sensation. We already mentioned the photoreceptors, but we also have internal receptors inside of our body, such as baroreceptors, that are going to detect the pressure inside of our body, and also different limb movements. Proprioception I find to be an interesting concept. This is the position of our body. So we don't actually have receptors for this, but we do track the movement of our body using our brains. So we do understand where we are in space based on proprioception. All right, so olfaction. We mentioned that these were chemoreceptors. Olfaction is done by a bipolar neuron that actually will grow and replace itself. So this is something that is actually frequently studied in science because we want to know how are these neurons constantly replacing themselves. And they actually grow through your nasal cavity. So raw nerve endings are there in your nasal cavity. And when a molecule touches that raw nerve ending, it will send a signal to the brain or to the olfactory bulb that then decides whether this information is important enough to send to the brain. So not much to be said about olfaction, except that it is a chemoreceptor and that it is a direct nerve ending. So there's no specialized cell. Taste is also a chemical sensation. We have many chemoreceptors on our tongue that we like to call taste buds, but not all taste buds are the same. We have many different kinds that each respond to their own stimulus or their own taste. For instance, sweet or sour or bitter. Now, there's no particular location on the tongue that is more sensitive to these areas. In fact, each taste bud usually has multiple taste buds on it. So for instance, the foliate papillae has multiple taste buds along the edge that will absorb that chemical tastant and respond to it. Each different type of papilla is different. For instance, the filiform papilla right here doesn't actually have any taste buds on it. It's mainly there for mechanical sense of the food, like the texture of the food that you're eating. As compared to the circumvallate papilla, which has the highest concentration of taste buds, but is actually really more sensitive to bitter flavors than other flavors. Even though it can taste all of them, it's most sensitive to bitter flavors. So learning about these different papillae and what they're specialized at is important, but all of the taste buds work in unison to give you that taste sensation. We are especially sensitive to things that are sweet because these are high energy foods, and also things that are very salty because we need that salt for our body. Now, we also have receptors in our skin. Most of the receptors in our skin are tactile, meaning that they are mechanoreceptors that are sensing the touch sensation on our skin. We have several different kinds. Learning the different kinds is important for the exam because you will be asked about these different types of receptors. But there are ranges. So we have Ruffini's endings, Merkel's discs, Messner's corpuscles, and Pacinian corpuscles. These are all detecting different types of touch. Merkel's discs, for instance, respond to very light touch. They're very close to the surface and they're almost like bare nerve endings, so they're very, very sensitive. Whereas Messner's corpuscle is more sensitive to things like vibration. Ruffini's endings will actually stretch, and so when they stretch, they fire or they signal that action potential. The Pacinian corpuscle does the same thing. It is there to detect deep touch, like pressure, and so when it is squished, it exchanges or it opens these channels physically for the ions to be exchanged, which will then fire an action potential directly from this nerve ending because it is encapsulated by layers and layers and layers of ions that when they are squished, they squeeze through the openings on this inner membrane and cause an action potential. So just the fact that you squeeze this nerve ending causes it to fire, I think is really cool. The Krauss-N bulbs actually are going to detect temperature more than touch. It's going to be sensitive to cold in particular. But if it gets too cold, then it will just start sending a pain sensation. So pain receptors are also called nociceptors. Nociceptor or pain receptors are throughout the body. And these are generally free nerve endings in the skin that will give you a pain sensation. And even if you don't have a nociceptor close to that part of your body, you can have something called deferred pain, where the pain feels like maybe it's coming from your arm, but the pain that you're actually experiencing is a heart attack. So that is based on nociceptors. But the thermoreceptors can also register as pain if the temperature gets too hot or too cold. We also have proprioceptors or baroreceptors that are going to detect changes in pressure inside of our vessels. And proprioception is to track and coordinate movement. Other animals have different kinds of perception. For instance, sharks and rays, and even some bony fishes, have electrical perception. They have specialized cells underneath their mouth or snout called ampullae of Lorenzini that are specialized in detecting electromagnetic fields. It's actually detecting its own magnetic field that the fish is producing itself, and then anything that enters into its magnetic field will manipulate that field, and the shark or the fish will be able to detect that change. So here they're not able to really detect this object because it's not interfering with its magnetic field, but the magnetic field that it's producing is being manipulated here, probably by this magnetic rock, so it would be able to detect that. We also have animals that have very, very sensitive tactile sensation. So just like we have tactile sensation using all of these different nerve endings, they have very sensitive nerve endings on their whiskers. And you can see that their whiskers are kind of organized in a grid-like pattern. Having them organized in that grid-like pattern gives them a lot of sensitivity, a lot of information as they're manipulated in different orientations. The animal can basically navigate the world around it in complete darkness because it can still have that sensitive touch sensation on these whiskers. So they were called vibrissae. They're tactile whiskers. Now, we said earlier that hearing is also a mechanical sensation, and I'll show you how. So sound waves are mechanical waves. They're pressure waves going through the air. The only reason they sound like anything to us is because our ear is interpreting those pressure changes as changes in pitch or volume. So the sound wave will go into your ear and it's funneled by your external ear. Your external ear's job is to funnel those sound waves towards your tympanic membrane or your eardrum. So the sound waves are going to be changing the tympanic membrane, which will then be caught by the malleus, incus, and stapes. These are called your ossicles. They're inner ear bones in your middle ear. These three little bones. So we just need to know that those are ossicles, and they are transferring the sound from the tympanic membrane to this round or to this oval window right here, the oval window. The stapes is tamping on the oval window. And what I find really interesting is there is a little bit of musculature here. So if the sound is really, really loud and it's vibrating really, really violently, that muscle will pull this incus away from the malleus, leading to a softer sound. Whereas if the sound is really quiet, those muscles can relax and cause the sound to become amplified, much louder. Now, what is the oval window? Well, the oval window is an opening to the cochlea, which is this snail shell shaped organ right here. And it is filled with liquid. Inside of that liquid are little hair cells that react to the vibrations from the sound. In that way, we can sense mechanically what we're hearing because of those little hair cells. Let's see another diagram. So here on this diagram, we can see the sound wave entering the ear. We can see the sound wave vibrating the tympanic membrane, which is in turn vibrating these three ossicles, which in turn vibrates the liquid inside the cochlea. The cochlea, if we uncurl it and straighten it out, it's just two really long tubes of liquid. And so the vibration starts in this end, goes all the way around, and then comes back out this end, and actually will exit out of the circular window, this round window right here. So the pressure is released, and that pressure can be sent down the eustachian tubes to relieve some of that. But what's interesting is the organ of Corti. The organ of Corti is in between these two canals filled with liquid, and they're going to respond to the vibrations of the liquid with these little bitty cilia, or little bitty hair cells. And their response to the vibration will be sent from our cochlea into our brain to sense hearing. The wavelength of a sound wave and the amplitude of a sound wave is corresponding to the pitch and volume. So how far apart these waves are, or the wavelength, corresponds to pitch. How tall the waves are, or the amplitude, is associated with volume. So we can change either volume or pitch with these sound waves, and your brain will recognize it thanks to the organ of Corti. So here in this diagram, we can see the two canals. It's this canal going this direction and this canal going this direction. We are just seeing it in a cross section, cut in half. So if I cut this right here, I would get this image. So we have the first canal, the second canal, and then in between the two canals, we're feeling the vibration. And there's this organ of Corti. It's this little pad that's going to be vibrating across the top of these little cells. And each of these little cells has something called sterocilia, or stereocilia, little hair cells that will respond to the vibration on this plate right here, the tectorial membrane. In that way, the pattern of vibrations can then be sent down our nerve into our brain and interpreted as sound. Your ear does something else though. Your ear also helps you with balance. So this part of the ear, the cochlea, helps you with hearing sound, but we also have these three semicircular canals. And these three semicircular canals actually help us with balance. Inside of these canals is a gelatinous material, and inside of that gelatinous material are otoliths. Oto meaning ear, lith meaning stone. These are little bitty crystals that are embedded on top of this gelatinous material inside of these semicircular canals. And look, here are those same hair cells again. It's the same type of cell, except these are connected to a different nerve fiber, so it's interpreted differently. These hair cells are going to give us information about the orientation of our head. So if your head is oriented normally, the hair cells will not be receiving any signals because the otoliths are static. They're not moving anywhere. But if you tilt your head backwards, the otolith moves due to gravity, brushing against these hair cells, telling your brain that you're oriented backwards. And this happens in 3 dimensions because we have 3 semicircular canals to give us all of that good information about our head tilting. And you can tell that this is going to be connected to our centers for balance and control. Other animals use their hearing differently than we do. We only use our hearing for sound, but other animals actually use it for navigation, called echolocation. Bats and whales both use echolocation, but they use it a little bit differently. Bats will produce sound in their vocal cords, in their larynx, and hear the sound with their ears. Whales, on the other hand, they're going to be emitting the sound from back here, and they're going to focus the sound through this oil-filled cavity called the melon. This is the melon. And then the sound will bounce off of whatever they're trying to see. They actually receive the sound in their very sensitive jawbone, in their lower jawbone. So whereas bats are receiving the sound in their ears, whales and dolphins are actually receiving the sound in their lower jaw. Um, we also have a different type of sense that humans don't have called magnetoreception that is primarily used for long navigation. So think about migrations, orientation of the animal to its surrounding environment in relationship to the weather in the seasons. And of course, animals do this differently, but all of them are going to have some sort of magnetic organ or a magnetic receptor, either in their beak or even their eyes. Some birds have these iron-rich crystals embedded in their retina. So they can literally see magnetism around them. Now, humans can see things with their eyes, but they are not, we can't see magnetic fields. We can see lots of different things along the electromagnetic spectrum as long as it falls within Roy G. Biv, or the visual spectrum, for humans anyways. Our eyes, due to the nature of physics, flip the image upside down because the image passes through a lens. Anytime an image passes through a lens, it will flip upside down, but that's okay because our brain will reorient it the correct direction inside of our head. So just because the image projected on our retina is upside down doesn't mean we go around seeing the world upside down. Our brain corrects it for us. Inside this visual cortex in the occipital lobe. But your vision also does something else. We have something called the optic chiasma. If you remember from the beginning of this lesson, the left brain is going to control the right motor functions and the right brain is going to control the left motor functions. Well, it's no different with the eyes. So the nerves for the eyes actually have to crisscross at something called the optic chiasma. So we're going to crisscross so that we get information from the visual field on both sides of the head. This means that your left eye is going to be sending the left visual information to your right brain, and the right visual information to your left brain, essentially. We're crisscrossing. This is interesting because when we have deficits in our vision, let's say that we had some deficit right here, it might affect our peripheral vision on one side versus the other. And again, we can tell the brain function and where these lesions are based on the type of vision problems that we're having. So if we're only able to see the left visual field, that may be a problem with the right side. Now, the eye works in an interesting way. Of course, we have the lens, and the lens is there to focus the light onto our retina. The retina is the reflective part of our eye. It's a thin layer on the back of our eyeball. And the lens can focus light because it's connected to this ciliary body, little muscles that can pull and stretch on the lens to help focus on different areas or focus on different depths, okay? Covering the lens is the cornea, okay? The front part is the cornea. And then in front of the lens is the pupil. This is just an opening. There's no actual structure for the pupil. The pupil is just an opening. So if you look through the pupil, you can actually see the retina, which is what happens when you have your eyes dilated. The doctor is pointing a light through the retina. Without it being dilated, he cannot see very much. So sometimes you'll get eye drops that will force your pupil to dilate so that the doctor can see more of the retina. Okay, but other than that, the light will go through the pupil because of the iris. This part is also muscular and also will contract to make the pupil smaller or relax to make the pupil bigger. Those little muscles right there. Now, the light is shining on the retina, but not all of the retina is really great at visual acuity because the retina is kind of built backwards. The reflective part is at the back, and then the light has to travel through all of the blood vessels, through all of the nerves, through the retina, and then bounce off of the retina and come back to excite are rods and cones. The rods and cones are what are actually seeing, or what actually determine your visual acuity. This is a picture of your retina. So imagine all of the light having to travel through all of these blood vessels and nerves. You're going to have some visual problems. So we have some different tricks to overcome that. One way that we have overcome that is by offsetting our optic nerve underneath the focal point of the retina. So the light, if it goes directly into our eye, should land on this area called the fovea. And your body has cleverly focused all of your rods, or at least, most of your cones in this area called the fovea, because that's where the light most likely will hit. Next to it, or right underneath it, is something called the optic disc. That's where the optic nerve comes in, and we don't have a lot of good visual acuity of that, because if you look at this image, there's a lot of nerve endings and blood vessels going through that area, so you're not going to see very well there. But there's not a whole lot of blood vessels or nerve endings here. Lots of visual acuity at the fovea. Now, I mentioned that there are rods and cones. The rods and cones, imagine the light going through all of these blood vessels, through all of these cells, reflecting off of the pigmented layer of the retina, and then exciting the rods and cones. That's crazy. Okay, so the cones are mostly going to be focused in the fovea, like this part of the eye right here. That's where most of your visual, or your color visual acuity, comes from. And cones contain a pigment called opsin, and there's 3 different types: red, green, and blue. Some combination of red, green, and blue is interpreted in your brain as all of the color spectrums. Rods, on the other hand, contain a pigment called rhodopsin, and it is much better at detecting movement and light and darkness. So you do mostly have rods. In fact, some animals only have rods. They're usually much better equipped for night vision. They're not as concerned with differentiating color as they are with running from predators or catching prey. So you can see that the cones are much smaller than the rods, and as mentioned, there's only 3 different types of cones, and there's 1 type of rod, and you see the rod is right there in the middle of everything, so it's really good at detecting light and darkness. The cones also just have a different shape. Other than that, they're going to be pretty much the same. They're going to have mitochondria, they're going to have a nucleus, they're going to have all the parts of a normal cell, but then they have this different part here that collects the light, and it contains the light detecting rhodopsin for the rod, or opsin for the cone. Here's a better image of it all kind of put together, where you can see the different rods and cones available, or organized here. All right, animals have different types of vision than we do. As mentioned, we only see a very small portion of the electromagnetic spectrum. A lot of animals can see outside of that electromagnetic spectrum, or outside of our visual field. Right underneath our visual field is infrared, or heat vision. We can't see it, but many animals can, such as rattlesnakes and lizards. They have organs called pit organs, usually located on their face or underneath their eye, that help them detect infrared light, or heat vision. Other animals have polarized vision. This is actually a side effect of them not having as much visual acuity. What they're actually doing is cutting out some of these sections so that there's a lot more differentiation between the colors. So they will see blue very vividly, they will see green very vividly, and they will see red very vividly, but they won't see the in-between colors. And the result is a polarized vision. A lot of people will use polarized glasses to go fishing, because as you can see, with the polarized vision, you can see clearly through the water, but with non-polarized vision, you see the reflection more easily than you see the fish, which I guess is about right here. So animal perception can be very different from your own. We also have internal receptors within our body that help us regulate homeostasis, such as baroreceptors that can detect blood pressure or the blood volume. We have chemoreceptors that help us detect carbon dioxide or acid levels in our body, and thermoreceptors that help us regulate our internal body temperature. So very interesting ways that we perceive the world around us. Especially, we need those nociceptors to detect pain. We do have internal nociceptors, but as I mentioned, that can lead to deferred pain. If we have somebody suffering from, let's say, a heart attack, they might feel that pain in their arm. Thank you for joining me for biology this year. I really enjoyed having you as a student, and I hope that you will join us for other courses in the future, but that is it for biology at LSCPA. I hope that y'all have a wonderful graduation, and I hope to see you succeed in the future. But don't forget, coming up, we have our Unit 7 exam, so don't forget to take it. And also, right after you take the Unit 7 exam, the final exam will open. While the Unit 7 exam is closed note, the final exam is open note, and you are able to use any notes that you took throughout the whole semester, but you are not allowed to use technology. So make sure that you have those written on paper. I hope that y'all have a wonderful summer, and come see me and tell me all of your success stories in the future.